Hello, my name is Anika Kuckertz and I'm a doctoral student in the Institute of Neuroscience and Medicine 1 in the Research Center in Jülich. And I want to talk about the multimodal. I want to talk about the multimodal mapping of the rat iso and proisocortex based on multimodal approaches. In the future, we expect to offer our multimodal parcellation schemes and furthermore the 3D data to the neuroscientific community through the HBP and eBrains infrastructure. As you can see here, a picture from the official HBP website. Within the HBP and eBrains infrastructure, multimodal tools for multi-scale brain mapping are offered, and they enable a great exchange of data between all the neuroscientists. And in addition to the investigations on the human brain, it is also necessary to understand non-human primates, but also rodent brains. As we all know, rats are the main used models in translational neuroscience or research. And including my study, I will focus on multimodal analysis, especially the cyto and receptor architecture in the rat brain. The topic of this year's Big Brain meeting is the relationship between brain microstructure and functional connectivity. As one main factor for neuronal signaling, the neurotransmitter receptors show heterogeneous, laminar, and regional distribution throughout the brain, which I want to show in that little piece of an area of the primary parietal cortex. I will explain later on how these colorful images are created. For the development of our comprehensive multimodal maps and parcellation schemes, the rat brain provides a good model because of the limited formation of gyri and sulci, which leads to a limited individual variability and therefore enhances the comparison between animals, but also between different receptor analysis of different receptor types. We started the multimodal approaches by processing 20 micrometer thin coronal serial sections of the unfixed rat brain in the cryostat. For each brain, around 1,200 to 1,400 sections could be processed. Afterwards, we use alternating sections for the visualization of cell bodies, and also for the visualization of one specific receptor type by means of the receptor autoradiography. Within this method, a tritiated ligand binds to the specific receptor type, and this could be visualized after exposition to tritium-sensitive film sheets. Here you can see an exemplary autoradiogram for the visualization of the muscarinic M2 receptor. And these autoradiograms were digitized as 8-bit gray value images, where darker gray values code for higher receptor densities and lower gray values code for lower receptor densities. Within these autoradiograms, the relationship is not linear, so we have to co-expose tritiated microscales with known receptor with known concentration of radioactivity in femtomole per milligram protein. And these microscales are used to compute a regression curve for the linearization of these autoradiograms. For visualization purposes, we assign equidistant ranges of gray values into discrete colors arranged in the spectral sequence from dark blue to red, which are indicated lower and higher receptor densities respectively. Till today, we generated comprehensive data of these 8 neurotransmitter receptors, which are shown in the table on the left side. And they all show different regional and laminar distribution patterns throughout the brain tissue. Today, I want to focus on the parcellation scheme based on the distribution patterns of the muscarinic M2 receptors. As you can see here, 2 alternating sections for the identification of cytohierarchy, tectonic borders, and their correlation to borders and for turbation of the borders of the M2 receptors. And we parcellate regions, for example, of the occipital cortex, either by a broader layer 5, but also by lower M2 receptor densities. And additionally, we identify higher M2 receptor densities in layer 4. And as another identification, the areas of the temporal cortex show the lowest M2 receptor densities. And that way, over 50 distinct areas within the iso and proisocortex could be identified along the whole rostrocaudal axis of the rat. By analysis of further neurotransmitter receptors, here, for example, kainate receptors, different distribution patterns could be enable more detailed parcellations. All these maps and parcellation schemes I have shown you up to now are in 2D, and of course we expect to bring them into the 3D rat brain volume. The M2 receptor autoradiograms were reconstructed and integrated into the waxome stereostactic space, which is the most widely used 3D reference space for the rat brain. Additionally, we plan to integrate our parcellation schemes into the waxome space and expand these maps by further neurotransmitter receptor analysis, which I've shown you before. And we want to make these data available for the neuroscientific community within the eBrains and HPP infrastructure. And coming to the end before lunch, I want to say thank you to my supervisors, and all the others are involved in the project and helped me a lot. And thank you for your attention.